red, blue, purple, green, off-white, pink. These all are the different colors which this Fabaceae plant has. And we will be trying to understand the ascent of sap with this same plant. This plant is commonly known as balsam, and we will be using this plant in order to study the ascent of sap. Now, when we want to study the ascent of sap, we have to also understand that we need a particular arrangement. And before understanding that arrangement, we should have a background knowledge of the position of xylem, phloem, and what are their functions. So let's see one small portion of root, stem, and leaf. We will start with the root structure. When we cut a small portion of the root and try to, and this is a cross section. So when we try to understand the different layers, we see that the outermost layer is the epidermis. Inner to it, the epidermis is brown color, whereas cortex is shown with yellow color, and inside it is presence of endodermis. After the endodermis lies the pericycle. I am talking from outside to inside. And after that, you can see the presence of phloem, and innermost area has the presence of xylem. So, what we can think of right now is we will be focusing only on the xylem and phloem. So, xylem is present at the innermost part of the root, whereas phloem is present out of or a layer outer to the xylem. So the core layer or the innermost layer is the xylem in the roots. What about the stem? In the stem, we can see the presence of cortex. There is presence of cambium, which is the triangular structure's outer end. The innermost part is known as pith, and sclerenchymatous tissue is used in order to or protect the cambium. So this is the arrangement, how there is presence of all these structures. Those blue colored, this structure which I am trying to point out, the edges are the ones which are known as xylem, and that band, dark band, is the phloem. So I'm talking about this particular structure, and the innermost one is xylem, whereas that black strip, I'm not able to point it out, it is the black strip. Yeah, this one is the phloem. So this is about the stem region. What about the leaf? In the leaf, the innermost again is the xylem, whereas the outer pink layer is the phloem. Xylem is represented by little purple shade, or I should say lavender shade. Different layers are shown: upper epidermis of the leaf, lower epidermis of the leaf, the presence of main vein. The central vein is there, the thickened portion. If you see a leaf, the most thick part of a leaf is the midrib area, and this is the midrib area where is the presence of xylem. Even xylem is distributed at different places, but this xylem at midrib is the one which supplies the maximum amount of water for the process of transpiration. And all the different layers, they are present in from epidermis till lower epidermis. So it will be, if we talk about from the uppermost part, uppermost layer is upper epidermis, then after palisade, mesophyll, spongy mesophyll. After that there will be presence of the lower epidermis layer, and in the spongy parenchyma there will be presence of various vacant areas as well. So this is all about different structures which or different positioning of xylem and phloem in different parts of a plant. Now only we will focus on xylem and phloem. If we are talking about the stem, the innermost part will be xylem, whereas the outer portion will be the phloem. So this is the only general idea which needs to be developed. You can see the pink portion. Pink portion is the xylem, the inside innermost part, whereas phloem is the layer which is outside the, or it is just the outermost layer which is present for xylem and phloem. Like demarcation is as such. Anyways, we will go into details now. Between the xylem and phloem, there is presence of one layer. That thin layer which I was talking about is the cambium. So if we want to understand this triangular structure, this is xylem, this is cambium, and this is the phloem. I hope now it is quite clear. So xylem, cambium, and phloem. Now we have an experiment. We have to understand one thing. What is it? That xylem transports the water and minerals, whereas the phloem transports the food. So what we have done is we have taken a plant which is balsam. From that plant we have taken out the outermost covering, that is the woody part is removed along with the phloem part, and we have intact only the xylem within it. So innermost part is the xylem. Our intention will be to leave the xylem and remove everything else, that is the outermost layer, phloem, cambium, epidermis, everything has to be removed other than the xylem, because we want to demonstrate that how is ascent of sap taking place. It means how is movement of water and minerals taking place in the plants. This is the experimental setup. We have one control, which is, this is one, this is two. One is the control, and what we have done is we have taken a stem, kept two stems, we have taken one, we have kept in one, whereas second stem we have kept in two. The leaves are still intact, and our intention is to remove two things. In the first setup, what we have done is we have removed everything very precisely with the help of a blade, and only the xylem tissue is left in the first experimental setup. Whereas in the second experimental setup, from the stem, we have tried to dig deep inside and take out the xylem and left the cambium, phloem, and every outer tissue as such. Now, we can see the same experimental setup after a couple of hours, say 5 6 hours or 24 hours. We will see that the first experimental setup, since the xylem is intact, the leaves which were there in the stem, they are turgid, they are completely open, and they can show every part of them, of their structure. Whereas in case of the second part, where we had removed the xylem, we see that the leaves, they have started wilting, or they have completely got wilted. The reason is the xylem is removed. Why do we call experimental setup 1 as control? The reason for that is that in this experimental setup, our xylem is intact, and we have to understand the ascent of sap only. So that's why it is the control. There will be no change in first experimental setup, and we will be observing the change only with respect to the first experimental setup. That's why it is considered to be the control. Now, what is the meaning of ascent of sap? The movement of water from the roots till the leaves with the help of root pressure, capillarity, transpiration, and various other processes which occur in the plant. Now, the most important two characteristics are there, or reasons are there for this purpose of ascent of sap. First is capillarity, which means a very narrow tube has a tendency to draw water molecules from the roots, or any fluid from the roots, or from very low area, or from a distant place to another distant place, just because the size of that tube which we are using is very narrow. And two another principles work in this process. One is cohesion, which means that the water molecules, they have a property of binding with each other because of hydrogen bonds being in between them. So this is cohesion. And another one property is there, which is adhesion. Because of adhesion, the water molecules, they bind to the xylem vessels. So when we think about both the properties together, it means water is attached to the xylem, water is moved attached to itself, like one water molecule with another water molecule. So as soon as one water molecule is removed, another water molecule will work as a trail. They will be flowing one after another, and this is how the entire process it works. Now, what about the root pressure? It is a positive pressure that pushes, that makes the cell sap move forward actively from the roots till the leaves. And over here, transpiration also plays a very prominent and important role. So these were the reasons for the ascent of sap, why the movement of water and minerals were taking place from the roots till the leaves. And what do we conclude? We conclude that the upward movement of water and minerals from the roots to different parts of the leaf occurs with the help of ascent of sap. And by the xylem, the entire process is facilitated. In the first experimental setup, xylem was intact. That's why the entire plant body remained turgid in spite of removing all the outer layers. Whereas in the second experimental setup, since the inside xylem was removed, that's why the leaves they wilted in spite of having everything else, the phloem, the epidermis, the cortex, everything was there, just there was removal of xylem. And that's why the plant it started wilting, or I should rather say the leaves they started wilting.